So I've just completed about a year ago a chronology of immunology, and I've called it molecular immunity, a chronology of 60 years of discovery. I had used to always tell the people in my lab that when I was all finished, I was going to write the history of immunology, because I figured that if I didn't write the history, I wasn't going to like whoever else wrote the history, and so I better do it myself. And so this book took me about 5 years to write, and I kept putting it aside because something else would come up and so forth. But I basically reread all of the literature from late 1950s until the present time. I referenced all of the papers that I thought were the most important papers. And so what has resulted is a compendium of references for the next generation of students, that if they really read all those papers, they'll learn immunology faster than their peers, essentially. And the book arose from the fact that one of the postdocs in the lab next door actually wondered whether he should buy a textbook of immunology. And I said to myself, or I said to him, I said, that's a waste of money. You shouldn't buy that. I said, first of all, you probably won't read it. And second of all, if you do read it, you're probably not going to understand it. And third of all, if you read it and understand it, you're probably going to forget it. And so you shouldn't do that. But if you really want to learn immunology, you need to read the primary papers. And so you have to go back in the beginning and read every paper in a prospective way. And so what we did then was we decided to do that in a journal club, so that once a week I would assign somebody from the lab a paper to read, and then they would present the paper together with slides and of the critical data and so forth, and we would make up our minds whether or not we agreed with what the author concluded or not, and why a particular paper was important in the context of the advancement of the science. So the first chapter I decided to start in 1957. And the reason being is that most of the history of immunology had basically already been written up until around 1970. And of course, 1960 was really the break point. And in actuality, it was really 1957, because there was an unbelievable paper that was published in 57 by Macfarlane Burnet, who is a physician from Australia, who had spent his entire career in investigating influenza virus and the immune response to influenza virus. And he did that from 1918 up until the late 50s. And he put all of his knowledge together in a hypothesis paper that he published in the Australian Journal of Experimental Medicine in 1957. And this hypothesis paper was only 3 or 4 pages. But in that paper, he basically set the stage and asked the questions that then a whole army of immunologists attacked over the next 50, 60 years. And the thing about that paper, and I recommend all students should read this paper, because if you want to become famous in immunology, this just shows you how to do it. You only have to write 3 or 4 pages. The thing is, he laid out, without any data and so forth, with all his experience, the major findings in immunology up until that point. And that starts then as a starting point for my book that started with him. And in his 57 summary, what he said is that nobody knew what the cells were of the immune system that mediated immune responses, that they could, since the beginning of the century and around the turn of the 20th century, people knew that if they injected things into animals or into humans, that there would be an immune response to those injected substances if the substances were foreign, like a microbe, a bacteria, or bacterial products, and so forth. And they, over the course of the first 50 years, they sort of mapped the course of the immune response, mostly discerned by antibody levels. Antibodies at the time, it was an activity noted in the serum that would react to whatever the antigen was, and they could devise tests where they could see that under the microscope or even with the naked eye. They could measure these antibodies as an antibody activity. But nobody knew where the antibodies came from in terms of which cells made them. And they didn't know what the antibody molecule, what the molecules were. The supposed is that they were molecules that were antibodies. But it took the first 50 years. It was only towards 1959, 1960 that the chemistry, the biochemistry of antibodies was finally solved. And they knew by then that antibodies were these molecules that look like a Y. And so on each end of the Y were the binding sites where the antibodies bound on to whatever the antigen was. Antigen being anything that would, it's a circular reasoning, anything that would activate antibody production is what they called an antigen. So by 1960, they knew that. And what Burnett proposed and hypothesized in his paper was that for the first time, really, that lymphocytes, which are one of the white blood cells, circulate in the blood, but also make up the major cells in lymph nodes and also in the thymus, that lymphocytes were really where the antibodies came from. And they did know that there were cells called plasma cells, and that had been worked out during World War II by a Swedish woman immunologist who traced the antibodies coming from plasma cells. But nobody knew where the plasma cells came from. All they knew was looking down the microscope, they looked different from the lymphocytes. But Burnett then said lymphocytes are the source of the plasma cells, and they are the cells that comprise the immune system. So that was really an intellectual breakthrough, essentially. And he went on to say that what happens when you inject an antigen into an animal or a human, that over time, over several days and so forth, that the lymphocytes that recognize the antigen, meaning that they have some means to find out that that's a foreign thing in the body, when they recognize the antigen, then they proliferate, and you get multiple clones of cells out of that initial reaction. And then he went on to say that after the what happens then is that those lymphocytes then somehow mysteriously deal with the antigen and clear it from the system. And then after that, after that, you're a different kind of a host, because now you've got this whole expanded population of cells that you didn't have before. And that he called clonal selection, a clone being an individual, a single cell, that when it sees its antigen, it just expands into many thousands and millions or more of exactly the same kind of a cell. So they're all like identical twins. So that's what a clone is. And so the theory that he proposed in this paper was called the clonal selection theory. And he went on to say then in this paper that people knew at the time that for the most part, people did not form antibodies and have an immune reaction to their own body, their own bodily components, cells and molecules and so forth. And so there was this phenomenon that had been demonstrated before 1957 that was called tolerance. So we had self-tolerance. We didn't react to ourself, but we reacted to everything that was non-self. And that became then the whole self, non-self paradigm in aspect in immunology that really defined what immunology, what the immune system was all about. And he said that the way that happens is that when the embryo is in the womb and it's growing and becoming an animal or a human and so forth, that cells, the lymphocytes, as the lymphocytes mature and so forth, if they recognize self, then those cells are eliminated. They mysteriously got killed by the body. And only the cells that recognize non-self go out and populate the body and circulate in the blood and go into the lymph nodes and so forth. And when they're out there, then what they do is just wait for antigens to come along that they can react to. So that really was a good explanation for what they knew at the time in terms of tolerance to self and reactivity to non-self. And so basically what he did by proposing this clonal selection theory is that he explained why vaccination works and why once, if you survive the disease like measles or mumps, or chickenpox or polio, if you survive that infection, then you have what they call immunologic memory. And that was explained by the fact that now you've expanded this whole population of cells that you didn't have before. Because those cells then persist, or at least some of them persist, for really, if it all works right, for the duration of your life. And of course, that's what happens in those after vaccination for those childhood infections. So that set the stage. And from 1960 until 1980 was really the golden age of what came to be known as cellular immunology. Because once Burnett told us what the cells were, then we started to look at lymphocytes. And soon, as soon as 1960, there was a paper published by Peter Knoll, who was a young assistant professor in pathology at the University of Pennsylvania. And he accidentally discovered that if you took white blood cells from the blood and incubated them with different kinds of substances, and he was interested in trying to make the cells divide so he could study their chromosomes. He left some in the incubator over the weekend, and he came in on Monday morning and he found that these cells were no longer the small, tiny lymphocytes that he put into the culture on Friday. And rather they'd become very big, huge cells. They looked like leukemia cells. And for a while he thought he'd discovered the cause of leukemia. But then as time went on, very, very rapidly, he realized that what these cells were doing was that they had been stimulated to proliferate by the substance that he put in with them, which was called phytohemagglutinin, which was abbreviated PHA. And that was the beginning of cellular immunology, because what he showed was that he showed a technique where you could take the cells out of the body. And before that, for the first half of the 20th century, immunology was all done in vivo, in the animal, or in the human, and consisted of injecting antigens into the body and then measuring for antibody levels. But once you could get these lymphocytes, and you could put them into tissue culture with sugar water and all the things that they needed in order to be able to survive and to grow, they would just grow. It was wonderful, and that created the whole field of cellular immunology, and it exploded. Just about everybody in whatever specialty of medicine that you can think about, they wanted to be able to do this too. And soon after 1960, other people said, well, I wonder if you can use that. Evidently, the PHA activated all of the cells. At least looked like it was all of the cells. And they wondered whether or not they could do the same thing, but with specific antigens, like mumps or measles virus or whatever. And so they tried it, and soon, within 1962, 1963, they could show that would happen. And then what the immunological community became very, very interested in this, and particularly, for example, the transplant people, because they could show that they could use antigens from donors to recipients and so forth and get this kind of a reaction. And so everybody started growing lymphocytes. And within a year or two after that, there was a new assay that was created by a fellow by the name of Niels Jerne, who was a Danish guy, who was actually a physicist, and he decided to become an immunologist and underwent a metamorphosis into that whole field. And he turned out he went on to win the Nobel Prize 20 years later. And one of the reasons he won the Nobel Prize was an assay that essentially proved Burnett's theory of clonal selection. And he was able to show with his assay that was ultimately called the Jerne plaque assay that an individual cell would make one type of antibody. And that's what Burnett had said. So along the line, just within a year or two after that, people started culturing these lymphocytes, and they found that if they took the culture fluid, that they grew their cells for a little while, a couple of 3 or 4 days. They took the culture fluid, and then they put that onto new, freshly isolated lymphocytes, that those cells would start to grow. And there were 2 papers that were published in Nature in 1965 that described this for the first time. And ultimately, what they described ultimately came to be called interleukin 2, as we'll get into as this series of videos goes on. And between 1965 and 1970, other people started reporting other kinds of what these culture fluids did to lymphocytes was to make them proliferate. But then with the Jerne plaque assay, other people showed that you could take these culture fluids and they would make the cells make more antibody. And then finally, towards the end of the 60s, they found that if you activated these lymphocytes and then harvested them from the cultures and then put them onto so-called target cells, cancer cells, whatever, that you could show that they would kill the target cells. And so that was those assays that were created in the 1960s and also in the 1970s became the basis for cellular immunology and gave the field the ability to start to dissect out what was causing all these weird phenomena that were going on. One of the things that happened as a consequence of that, as time went on in the 70s, is that they found they needed several different kinds of cells in there, not just lymphocytes. One of the first that they found was they needed cells that were called macrophages. And what macrophage means is macro means big and phage means eater. Macrophages, what they were found to do is that if you put foreign substances in with them in culture, that they would engulf them. They would phagocytose this material, and they would then digest it, break it down into the individual components, molecules, and then they would. That's one of the ways the immune system would rid the host of the antigens. Well, it turns out lymphocytes, they knew by then that lymphocytes became plasma cells which made antibodies. There was this other kind of cell there that ultimately became called a T cell. And around 1965, Max Cooper expanded on experiments that had been done by Jacques Miller, who had been studying the thymus. The thymus is a gland, is a collection of lymphocytes that sits in the chest above the great vessels as they come out of the heart. And so they could take this out of animals and also humans and look at it under the microscope, and all they saw were these very small, sort of resting lymphocytes. But they did a bunch of experiments, and they found that none of these lymphocytes would make antibodies. So they said, well, what is that all about? Are they really lymphocytes and so forth? And to make a long story very short, over the course of the 1970s, they found out that those cells, because they came from the thymus, were called T cells, and they were lymphocytes. And the cells that became plasma cells that made antibodies came from the bone marrow. And so those were called B cells. So the B cells were precursors of antibody-forming cells, and the T cells were found that they had to be there too. You couldn't just put the B cells into culture and have them make antibodies. And so then the question was, how do the T cells do that? And the T cells were found to make funny molecules, funny activities, and release them into the culture medium. And so ultimately, there were, around 1975, several different kinds of T cells that had been discovered and described and characterized. And so people started understanding that there were helper T cells that would help the help the B cells make antibody. But there were also a separate subclass of T cells that became killer cells. And so those were called cytotoxic lymphocytes, cytotoxic T cells. And then the macrophages. And in order to put cells into culture, to make antibody, or to become really good killers, you needed all three of these different kinds of cells. You needed the B cells, you needed the T cells, and you needed the macrophages. So then the questions became, as that decade went on, the questions became, well, how do they do that? What happens? And so assays were invented by different workers to assay for the activities in the supernate. And so we went from, in the first half of the century, measuring activities of what became antibody molecules. And in the second half of the century, we started measuring activities that the T cells made and the macrophages made. And that whole thing took another decade until the 1980s to become, so that things were characterized and so forth, and it was reasonably clear what was going on. And during that time, in the late 1960s and also in the 70s, people found that you needed to have histocompatible cells. In other words, if you mixed my cells with my neighbor's cells, it wouldn't work. You couldn't get antibodies out. That was the first thing that was discovered. And then other people showed that you couldn't generate cytotoxic T cells either unless you had histocompatible. You couldn't get proliferation and the generation of cytotoxic cells unless you had histocompatible, meaning from the same person or the same strain of mice is what was used primarily. And nobody understood that. Why was that the case? And it was basically that focus by late 1970s, early 1980s, that had focused the field. Is the primary question was particularly how the T cells recognized the antigen, because what they found was the T cells only recognized antigen that was on the surface of other cells, whereas B cells had on their surface, they had surface antibodies, and those would recognize antigens just floating around in the medium. And so you had this sort of dichotomy and this conundrum of B cells and T cells in terms of how they recognized the antigen. And so towards the end of the 70s, there was this fellow by the name of Baruch Ben Asraf, and he really ultimately he published another opinion piece, just like Burnett had in 1957, that really was put everybody on the right track. And what he hypothesized was that the antigens on the surface of a cell that the T cells recognized were either put on the surface of the cell independently, but in addition, you needed another molecule that must have been coming from genes that coded for the histocompatibility between different tissues. So what evolved was a two receptor hypothesis that there was antigen, and then there was what they ultimately called MHC, which stands for major histocompatibility complex. And that was the name for the gene. Or the other alternative is that the antigen actually bound to the MHC coded molecules. That was the one receptor hypothesis. Well, and so Banasarab hypothesized that this is the way the whole system was working. He didn't really solve the problem between one receptor and two receptors, but he said that the histocompatibility was very, very important, that it had to, and that's how he explained it. And so he went on then to win the Nobel Prize in 1980, basically for that hypothesis paper, although he had also a lot of experimental data that went along with it, as well as others. So then, as time went on, in the early 70s, people started looking at what the cells were making in these supernatants, or in these culture media, and particularly the macrophages seemed to be very, very intriguing. And it was found early on that macrophages would release some kind of a soluble activity that would markedly enhance the ability of the T cells to proliferate once they recognized their antigen. And that activity was ultimately called lymphocyte activating factor. And it was shown that macrophages released this thing, and that they would respond to different kinds of stimuli that were known already to be very potent macrophage stimulants. And so that was out there. But then, in addition, there seemed to be T-cell derived activities that were coming out of T cells that were also allowing or promoting the ability of the cells to proliferate. And so multiple papers were published between, say, 1965 and 1975 with multiple different names: T-cell activating factor, T-cell stimulating factor, T-cell replacing factor, all of these different kinds of things. And nobody could really sort out what it was that was involved in all of this business. And so that really brings us up until close to the end of the 70s and into the 80s, in terms of what was known about how the immune response, who was doing it and how they were doing it. And it was clear at that point in time, around 1980, what you needed to do is you needed to identify a molecule or molecules which were the basis for these weird activities that were going on. You needed to purify it. You needed to generate huge quantities of it so that you could study it, and so forth. And so that began then the next era, which we're still in, which is what I call molecular immunity. Because what we were able to do in 20 years, from 1960 to 1980, was to go from, before 1960, we were dealing with whole animals. And then between 60 and 80, we reduced the immune response to cells in culture media. And then after 1980, it was clear that what needed to be done was to identify all these molecules. And what that did was then went from a macroscopic situation, sort of a macroscopic meaning that you could see it with the naked eye. And so you can see the width of a human hair with your naked eye. That's about it. That's 1 millimeter, essentially. So a cell is 1,000 times smaller. It's 1 micrometer, or 10 to the minus 6 meters, whereas a thousandth of a meter is a millimeter, and that's 10 to the minus 3rd meters. 1/1000th of a meter, and then 1/1,000,000th of a meter. Well, molecules are 1/100000th of a meter, so you had to go to 10 to the minus 9. And that transition that occurred like in 1980, going from 10 to the minus 6 to 10 to the minus 9, was really a revolution. And really a revolution in all of biology, and biomedical science, and it really rested upon several discoveries that occurred in the 1970s, one of which was genetic engineering, gene cloning. Because up until that time, when investigators wanted to find out about a given molecule, they went to the slaughterhouse and they got kilograms and kilograms of, say, beef heart. And they'd take it back to the lab, and then they would grind it all up, and then they would start to separate it with biochemical separative procedures. And what would happen is that you'd start out with kilograms, and you might end up with 1 microgram at the other end of your purification process, which wasn't enough to really do much with, except to say, well, this is what it is. But you then couldn't take it further. So gene cloning allowed us to get from the cells to the genes, and from the genes, then we could get to the gene products. And the gene products, basically, and I'll go into that later, were the proteins that everybody was interested in. That was one discovery. Second discovery in the 1970s were monoclonal antibodies. Turns out these were equally as important, because what that allowed us to do, and which was really another solidification and confirmation of Sir McFarlane Burnett's clonal selection theory, because what you could do is that you would get clones of lymphocytes to make a single individual antibody molecule that reacted to whatever antigen you wanted. And so that's what a monoclonal antibody is. But because of the fact you grew the plasma cells in the culture that were making these antibodies, you could make unlimited quantities. It's just a matter of how big your culture vessel was and how many, and so forth. And so so the genes allowed you to make a lot of the protein, but it was always unpurified. It had all kinds of other proteins in there too. But now with monoclonal antibodies, you could pick it out of the soup and purify it and isolate it and show through different procedures that this was only one molecule there. Given that, then, if you then went one step further and then you took your purified molecules that you convinced everybody that was really pure, and then you could do very definitive experiments. And this is basically what this is called as reductionist science, which is what we've all been doing from the beginning, going from the total animal down this, reducing it down in size and complexity so that we could study it and characterize it.